Hello and welcome to the WeXG Online Academy. My name is Chris and I'll be your tutor for today. In our previous video, we looked at questions 31 through 35, and in this video, we'll look at questions 36 to 40 of the Cambridge As Biology Paper 1-1, Winter 2019, course code 9700. Question 36. Outbreaks of cholera commonly occur in refugee camps that are set up after major natural disasters such as earthquakes. The list shows some measures that can be taken to limit the spread of cholera in the refugee camp. Which of these features control measures involve an economic factor? Okay, so let's have a look through the ones and see. So treating all drinking water supplies with chlorine. Well, yes, this requires an economic factor. You must provide the chlorine to be able to do that. Setting up emergency treatment center to isolate cases of cholera and treat them with antibiotics. Yes, there's an economic factor there. You have the cost of infrastructure and also the treatment of medicines. Using concentrated disinfectant to clean sewage disposal areas and infecting bedding. Okay, yes, this also is an economic factor. You need money to be able to purchase disinfectant, and then you've got the labor needed to clean these areas as well. Health workers visiting regularly to detect cases. Yes, this is also an economic factor. You must pay the healthcare workers to do this. Number 5, keeping good records and numbers of cases of deaths at treatment centers. Yes, you need administrative workers to be able to keep accurate records of all these cases, to be able to map and monitor and therefore hopefully prevent any further spread. So option A is the only answer that includes all 5 of them, okay? So we can eliminate B, C, and D because all of these are suitable factors that need economically be involved with, okay? Question 37, which use of antibiotics help reduce the spread of resistance in bacteria? Okay, well, using a high concentration of that antibiotic to kill the bacteria doesn't really reduce the spread of it. It does increase the chance of successfully killing the bacteria, but it doesn't reduce the spread of resistance as such. Okay, so option A is incorrect. B, giving routine preventative antibiotics to people having an operation. Well, again, if I keep giving people antibiotics, this means it increases the chance that maybe some of them might mutate and therefore lead to an increase in resistance. Okay, option C, regularly changing the type of antibiotic used to treat particular bacterial infections. Yes, this is a potential way of reducing the spread, okay, because at this point, any bacteria that survive, there's less likely to survive if I use multiple antibiotics for the same disease, okay? If I keep using the same one over and over and over again, then some bacteria might mutate and therefore lead to the mutation of resistance occurring. So if I change the treatment regularly, it should prevent the spread of bacteria being resistant to a particular antibiotic. Okay, option D, although this is a method of treatment of giving antibiotics to low-level infections, it doesn't necessarily reduce the spread of it. Okay. Question 38, the graph shows the amount of antibody produced in response to an antigen. From the graph, which statement is correct? A, it takes 25 days to achieve active immunity. Well, when your body first started making its antibodies, around about this day, okay? So at this point, this is where you start having your active immunity. However, as it's a primary response, there is a high chance that you would suffer the symptoms of the disease, and if it's a fatal disease like tetanus, you could die before your body's memory cells are created to mass produce these antibodies. So option A is incorrect. Memory cells for this antigen are present in the body within 20 days. Well, yes. Okay, during this time, the cells, the lymphocytes have had time to figure out which antibodies to create, and as a result, the concentration decreases. But as this point goes on, memory cells has been made for when you have a secondary exposure and a mass increase in concentration of antibody taking place, leading option B is the correct answer. T lymphocytes are activated on day 12. No, that's incorrect. The helper cells are releasing cytokines and other sort of substances to help, but day 12 is not the day that they are being activated. The second exposure on the antigen occurs on day 25. Well, day 25 is roughly in the middle, and as you can see here, this is where the exposure would have taken place, leading to a mass increase. So it's on about day 22. So option D is also incorrect. Question 39, which statement summarizes the hybridoma method for production of monoclonal antibodies? Okay, so what would normally happen is that you would inject a mouse or a small mammal with a particular pathogen or antigens of that pathogen. Okay, with that in mind, we can eliminate option A and B. We're not going to inject them with antibodies because that would create a passive immunity, and we want an active one where the mammal is actively producing the antibodies for a particular antigen. Okay, now we don't harvest antibodies from the blood because here we want our cell to be able to make its own antibodies. So what we do is we then harvest the B lymphocytes from the spleen of, say, said mouse. We'll use those B lymphocytes, we'll fuse them with a cancer cell because cancer cells can divide rapidly, and then what would happen is we would test for the antibody to see if it's combined and successfully mass producing that antibody, and then if it's successful, we will then produce this on a very large scale. So this supports option D as the only correct one. We eliminated C because the antibodies themselves won't be able to be reduced as an antibody. We would need a cell that could physically produce them. And it's the B lymphocytes that actually do that. Okay. Question 40. The vaccination program for smallpox was a medical success story which resulted in the World Health Organization finally declaring the world free from smallpox in 1980. Which statement correctly identifies the reasons for the success of the smallpox vaccination program? So the virus infected animals were making it easy to stop the transmission cycle. Well, that's not true. The vaccination is not preventing the transmission. The virus remained in the body following the infection and then it became active later. Well, that's not true. Otherwise, if it became active later, it still would be a problem in modern society. The virus was stable and not prone to mutations. Okay, well, if this was the case, C is the only option because if the smallpox virus did mutate, it would need multiple vaccines for that. Okay, however, the smallpox virus has not mutated and therefore the antibodies that are specific for that are successful within every single vaccination that are given to it. Okay, option D is incorrect because if it was unstable and prone to mutations, it would lead to multiple infections occurring yearly, like the influenza virus, which can mutate readily, and therefore it would stimulate primary responses in lots of individuals every time they're infected, and there'd still be a large percentage of deaths each year due to this disease. Thank you for watching our video today. Please register on the website for a free demo, and I look forward to seeing you in our next video.